Now let's take a look at the structure of nitrogenase. And so recall we looked at a schematic, which was shown here again, of nitrogenase. And when we talk about nitrogenase, we're actually talking about two different proteins. So the first one is the iron protein. Okay, and the schematic is shown here. This is where there's ATP binding and hydrolysis. And then there's the MOFI protein, which is shown as these rectangles. And that includes the active site, which is called the iron molybdenum cofactor. And so we'll talk about this again in a second. And so what happens is, remember, this is the specific reductant for the MOFI protein. This iron protein is the specific reductant for the MOFI protein, and this shuttles electrons to the MOFI protein, and that eventually transfers that to nitrogen. So what happens is this is only bound, the iron protein is only bound to the MOFI protein when, there's ATP bound to the two ATP binding sites, and there's a cofactor in here called iron sulfur cluster. And when that's reduced, that will allow for binding to the MOFI protein, but only under these cases. As soon as the ATP is hydrolyzed, there's a very rapid dissociation of the iron protein from the MOFI protein. So it's really difficult to get a good picture from a crystal structure of this, but it has been done. And this was done by using a transition state analog of hydrolyzed ATP. So in this crystal structure, ADP ALF minus was used as this analog in the ATP binding site, and that allowed for trapping the nitrogenase, the full nitrogenase complex of both the iron protein and the MOFI protein. So let's take a look at each of these subunits. So we're actually looking at two separate nitrogenase units, if you want to think about that. Okay. So in the center, interacting with each other in this crystal structure, are the MOFI protein. Okay. So the MOFI protein for one of the nitrogenase subunits is shown in red and this aqua color, and then the other MOFI protein is in this red and blue. Okay, and those are the two. So each of these MOFI proteins has two subunits. So this is a heterodimer. Each of these is a heterodimer. Okay, so one subunit, for instance, here is colored blue, and the second subunit is colored red. All right, within here, there are two cofactors called the P cluster and the FeMo cofactor. Okay, the P cluster shuttles electrons from the iron protein to the FeMo cofactor, the iron molybdenum cofactor, and the iron molybdenum cofactor is the active site of nitrogen reduction. So on the outside of these are docked the iron protein. Okay, and again, as we discussed, this is the site of ATP binding and hydrolysis. There is an iron sulfur cluster within this iron protein that shuttles electrons one at a time to the P cluster and then on to the iron molybdenum cofactor. All right, and then this is the full complex of the nitrogenase. So you can think of this as two nitrogenase units that are interacting with each other within this crystal structure. And again, this one, the MOFI protein is a dimer, a heterodimer, and then the iron protein itself is a homodimer. Okay. And those are colored by green and pink. Each of those subunits is separate subunits is colored as green and pink in here. Let's take a closer look at the redox cofactors that are involved in this. So as we noted before, redox processes and electron transfer are very important for this. So here's a nitrogenase subunit with the iron protein on top. Okay, this is the initial site that gets reduced by a ferredoxin. The 4-iron 4-sulfur cluster is the cofactor within the iron protein, and this shuttles between a +2 and a +1 oxidation state. So in the reduced state, the iron sulfur cluster is in the +1 state. In the oxidized state, it's in the +2. So this ultimately takes the electrons from either photosynthesis or oxidative respiration. This brings the electrons to the MOFI protein, where there is a P cluster, okay, which looks a lot like a 4N4 sulfur cluster that shares one sulfur in the center. Okay, and then the electron is then shuttled to the P cluster, and then finally to the active site, which is called the iron molybdenum cofactor. So let's take a closer look at the iron molybdenum cofactor, because this is a very unique cofactor within biochemistry, within biology. Okay, and so there's a close-up of the iron molybdenum cofactor, and so you can think of this as one. There's a 4-iron-3 sulfur cluster on the left-hand side here. All right. On the right-hand side, it is connected to a molybdenum 3-iron-3 sulfur cluster. So in orange are the irons, 1, 2, 3, and then over here is a molybdenum. Okay, and so attached to the molybdenum as well is a homocitrate ligand, which is shown here for a schematic is shown here. And then in the middle of all this, what is bridging the two metal sulfur clusters, all right, is this atom, which for a long time was unknown, but was postulated to be either oxygen or nitrogen. But this has recently been found to be carbon. And this was determined by a combination of high-resolution X-ray crystallography and also spectroscopy, and done in two separate papers or two separate studies that confirmed that this is a bridging carbide in the middle of this iron molybdenum cofactor. So it's a very interesting feature, in addition to the already interesting feature of molybdenum within this cofactor, which is not a common metal amongst metallic cofactors. And so this is the site of the nitrogen reduction and of nitrogen reduction to ammonia in nitrogenase.